Hi, everyone. Just wanted to do a quick little final review for your final exam that should be taken this week. Just a little housekeeping. It's finals week, so you have to complete the final exam in order to pass the course. If you don't take the final, you get a 10% overall final grade. So you want to make sure to take your final no matter what. You want to schedule it with your site coordinator if it's proctored. If you're unsure, then check your folder to see which one, or you can ask me and I'll let you know. Unit folders, all of them, all 8 are still open, and hopefully you can catch up on some missing work if you have any. Class ends Friday by 5 p.m. Unless you filed an extension, then it cuts off at 5 p.m. So make sure you have everything in. You can still get an extension, but you have to apply as soon as possible. So far, I only have 1 student. This is in the yellow folder, the end of course items in your unit view. It's towards the end, just above unit 8. And also I'm giving out extra credit if you fill out the survey for 25 points. There are 8 units: 1 scientific method, 2 water and biochemistry, 3 was cells, 4 cellular respiration, 5 was photosynthesis, 6 was cell division, 7 was genetics and heredity, and 8 was DNA to protein. Scientific method and study of life was our first one. The definition is it's a plan for asking questions and testing possible answers. It generally follows the steps listed in the image to the right. So it starts off with some sort of observation, things that we're seeing happening. And then from there, we wonder kind of like what's going on. Can we ask a question like that? Then we start to do a little research, figure out what's happening. So we're going to predict what's happening. And then the fun part is testing out to see if we can get the answer to it or not. And we draw our conclusion, we accept our hypothesis or decline our hypothesis, and then we communicate our results. Metric conversions, you do have a problem or two that are asking you to convert things. Remember in this video in the lesson, there was a trick. So King Henry died by drinking chocolate milk. So the letters K, so that would be kilo, H, hecto, deca, deci, centi, and milli, depending on where you're at. So for example, on number 1, 56.3 centimeters, it's centimeters. So you go over to the seed, second to the right. And then in order to get millimeters is the next one to the right, you're just going to move the decimal 1 place to the right. Milliliters compared to liters, milli. So you would end up moving it 4 decimals, 1, 2, 3 decimals to the left. And again, so you have to keep the King Henry died by drinking chocolate milk all in order for it to work. Check out that video if you need a little extra help before you take your final. 7 characteristics of living things. There's a lot of details in this, but basically they have to possess 7 characteristics, and that's they are to animate or living, ability to respire, grow, excrete, reproduce, metabolize, move, and be responsive to the environment. A big part of this is nutrition. So can they take it in, absorb? Plants do photosynthesis. Excretion is removal, so waste removal. So that's toxic materials, waste products, substances in excess of, or like chemical reactions. Respiration is breaking down food, so that's food and cells, and then releasing energy. Sensitivity, do they change to their environment, stimuli? For example, like our pupils when we go indoors and outdoors. Reproduction, can they reproduce? Overall, the goal is to prevent your species from extinction. Growth, so they can increase in size and mass, or the number of cells. Or cell size. Movement, is there changes in their position or their place of an organism or part of an organism? For example, like our hands. Parts of plants actually move really slowly to get more light. Leaves, they always grow towards light. So if you flip a plant upside down, then it would eventually kind of come back up and try to go towards the light. There's levels of organization. This is just more of the organism cells. This is not ecosystem one, but you could look at that one too. But cells, a basic structure and function of all living things. That's your smallest level of organization. The next up from there is tissue. So a bunch of cells make up a tissue. Then a bunch of tissues make up an organ. A bunch of organs make up an organ system. And then a bunch of organ systems make up the organism. Next unit was water and biochemistry. Basically, with water, we talk a lot about covalent bonds and cohesion. A covalent bond involves the sharing of electrons from different atoms that form electron pairs. So if you've looked at chemistry at all, this is familiar to you. Hydrogen and oxygen share electrons right there, and it creates this really strong hydrogen bond. Cohesion is just when water molecules are really attracted to each other. And again, that has hydrogen bonds on there because hydrogen bonds are really strong. And cohesion is attraction between molecules of the same substance rather than adhesion. There's chemical compounds needed by living things to survive, and we've learned and talked about in a discussion board carbohydrates, proteins, and then lipids, which you'll also see as fat and oils. So you need to make sure that you know the difference between a carbohydrate, a lipid, and a protein. Cells, we looked at parts of a cell, plant and animal cells. Organelles are tiny, like organs of the cell. The presence of organelles is one of the distinct features of a eukaryotic cell, so you want to know that. The nucleus, you know, the kind of brains of the cell, the mitochondria and the energy part. The cool thing about plant cells is they have these chloroplasts that are kind of like stacks of green pancakes, and that's where they convert their light energy to light into their energy. So you need to know the difference between prokaryotic and eukaryotic cell. Prokaryotic are very tiny. They have usually flagella. They have ribosomes in there. They do have a cytoplasm and a cell wall, but their genetic information is kind of floating around in the center, whereas a eukaryotic cell has your membrane, mitochondria, Golgi apparatus. The nucleus is the big difference. All cells have genetic material, cytoplasm, ribosomes, cell membranes, and cytoskeletons, but prokaryotes, they don't have a nucleus. They have the DNA just kind of floating around in there. They have some ribosomes to make proteins still, but not any of the major ones, where eukaryotes have nucleus where the DNA is in it, and then they have a bunch of different organelles. They also have ribosomes for protein synthesis. You need to know active versus passive transport. They're two important biological processes that help move substances such as nutrients, oxygen, water, and other macromolecules within the cell along the elimination of waste products. So waste has to travel in and out of cells, and that's super important. Passive transport does not require energy, and that's the big thing. So molecules flow across membrane by diffusion, whereas active transport, it requires a lot of energy, and they're actually pumped across the membrane. Cellular respiration was our fourth unit. I would say probably one of our hardest units for sure. Cellular respiration is a set of metabolic processes. The cells break down the glucose molecule to convert its stored biochemical energy into adenosine triphosphate, which is ATP. It occurs within the cells of all living organisms, including both prokaryotes and eukaryotes. Cellular respiration is categorized as an oxidative process because an electron donor is oxidized here and oxygen gets reduced to produce carbon dioxide, water, and energy. So this is a vital process providing all the energy to maintain life. There's two different types of fermentation. There's alcohol fermentation and lactic acid fermentation. It gets energy from glucose and does not require oxygen. Again, there's the two different types. Alcohol fermentation is your alcohol, bread, wine, and lactic acid fermentation is actually in our muscle cells, like when we work out. So if you work out and the next day you're really sore, that's lactic acid. There's aerobic and anaerobic respiration. Aerobic literally has the word air in it for a reason. It takes place in the presence of oxygen. That's that green little side there in the mitochondria, whereas anaerobic takes place in the absence of it. And if you don't have air, then that's when things ferment. Unit 5 was photosynthesis. The definition is process by which green plants and some organisms use sunlight to synthesize food from carbon dioxide and water. Photosynthesis in plants generally involves a green pigment chlorophyll and generates oxygen as a byproduct, which is really good for us, and it also recycles carbon dioxide, which we breathe out. The equation's down below, and then there's a really good picture to the right. We need to know glucose, chloroplast, and the thylakoids or the granum. And thylakoids are the membrane brown structures embedded in the chloroplast. Stroma, the stack of thylakoids, is called a granum. It resembles a stack of coins or green like I think of them green pancakes. Chloroplast is the organelle in which the cells will plant and is in the site of photosynthesis. And glucose is a simple sugar in which an important energy source in living organisms and is a component of many carbohydrates. It's used by plants for energy and to make other substances like glucose and starch. There's a lot of differences between these two, but there's also a lot of similarities. ATP is involved in proteins, provides energy for cellular functions. It uses ATP synthase. It uses electron transport chain, and it's electron carrier. So those are some similarities. Our photosynthesis is, it needs light. Energy is trapped. It converts carbon dioxide and water into oxygen and glucose, which is important. It's anabolic, where cellular respiration can be. It's a catabolic process. It converts oxygen and glucose into water and carbon dioxide. So they flip-flop right there. And cellular respiration occurs in all living things, whereas photosynthesis is just in plants. 6 is cell division. We have the cell cycle, the process through cells replicate into 2 daughter cells within the same genetic material. There's G1, S, G2, and then our M, which is mitosis. It's a process of cell duplication or reproduction during which one cell gives rise to 2 genetically identical daughter cells. Meiosis is a type of cell division in sexually reproducing organisms that reduces the number of chromosomes in the gametes, which are the sex cells. Independent assortment. This is something you need to know for your final. It's during the gamete formation, different pairs of alleles segregate independently of each other. And so you, in this parent cell, for example, you have capital A, capital B, little b. So which allele a gamete receives for gene A has no bearing on the allele it receives for gene B. So it's produced evenly in meiosis through all of them. So that's a really good example that you can see there. And again, that's kind of relates to your Punnett square. So if the circles are kind of confusing, you think of Punnett square. Unit 7 is genetics and heredity. Again, our Punnett square is super important to identify possible allele combinations, whether they're homozygous or heterozygous. And just for the record, homozygous is the same letter. So they're both either capital or they're both lowercase. Heterozygous is different, so a capital and a lowercase. There's a lot of different genetic disorders that are caused by structural changes to a chromosome or excesses of deficiencies of entire genes located on chromosomes. So a really good example of this is trisomy 21, which is Down syndrome. Actually has 3 instead of 2 chromosomes there. And then genetics is the study of genes, heredity, and genetic variation in living organisms. The probability is a big one, and I feel like that's something that you guys can look at more to better understand Punnett squares and ratios. So the definition of probability is the likely outcome that an event will occur. We use that to predict the chances of gametes and offspring having certain alleles. So here's a Punnett square. Another thing to remember is genotype is the genome, whereas phenotype is a physical characteristic. So phenotype starts with a P, that's physical appearance. So brown hair, blue eyes, etc. Whereas genotype is more of your lettering in this case. And again, where you guys kind of get confused is we're looking at ratios. So our possible ratios are 0 to 4, 1 to 3, 1 to 2 to 1, and then reverse of that. A capital letter, anytime you see it, is dominant. So if there's a capital letter, they will see the dominant trait no matter what. It's only unless they have the little lowercase that will receive a recessive. So where it looks like both parents are carriers of a gene, so capital A, little a, that's heterozygous. And there's only a 1 in 4th chance that they'll be recessive. So 3/4 chance, so 75%, they'll have the dominant characteristic. There's a 25% chance that they'll have the recessive. Example of this is like brown hair and blonde hair. So blonde hair would be lowercase a. It's a recessive gene. Here's another good example. Huntington's disease is an inherited disease caused by a dominant allele. So when a parent has to have a dominant allele, the Punnett square shows the genotypes of a male and female and the predicted genotypes of their offspring. What is the missing genotype? Again, genotype is here. So one of them has a capital H, lowercase h. And you can see that right below the mother. And then the father, they're saying, is lowercase h and lowercase h. So when you look at that, there's basically a 50-50 chance that they're going to get Huntington's disease because the mother is a carrier, whereas the father doesn't carry it at all. If the father was a carrier and the mother was a carrier, then there'd be a 75% chance that that child would have Huntington's disease, and then a 25% chance that they wouldn't, where the little h's are. Our last unit, DNA to protein and biotechnology. We looked at DNA replication and DNA synthesis. It's the process of copying a double-stranded DNA molecule. It's that helicase shape that twists like a twisted ladder. It involves copying the genetic material and passing it on to daughter cells. Therefore, the process is very important to continuation of life. Basically kind of unzips and then the other sides get replicated. A gene mutation is defined as an alteration in the sequence of nucleotides and DNA and a polymer of nucleotides joined together. So anytime there's a different sequence that's not supposed to be there, that can be a good or bad mutation. A lot of the time I would say it's not good, but sometimes it does help you. Transcription and translation is how we make proteins. Super, super important to know that. Here's another question that you'll see is the central dogma. The central dogma of a molecule is a theory stating that genetic information flows only in one direction. So it either goes from DNA to RNA to a protein, or it can go from RNA to a protein. You can have reverse transcription from RNA to DNA without replication, but it always goes from DNA to RNA to protein, or RNA to protein. All right, good luck on your final. Let me know if you have any questions. My cell phone number, feel free to call or text me. I'm pretty responsive to that at any time. And if you're watching this online for extra credit, your word is photosynthesis. So email me photosynthesis and I will give you extra credit points. All right, thank you.